All right, so we're going to look at the cell first. First, look at the first PowerPoint here. So let's take a look here to see the cell model. Now, there's going to be some description coming up, but on the first lab exam, you'll probably see questions that relate to identifying these structures, and there'll also be questions that relate to the function of these structures. So you have to be able to do both, identification and function. And either pictures or multiple choice or just purely some structure. And you could either write plasma membrane for that or cell membrane, okay? And you need to be as specific as possible. The choices really are very specific. We just want the direct answer. There are no sentences that we want, just 1 to 2 word answers, nothing more than that. Otherwise, it'll probably mark it wrong off the bat. So what's coming up is you will be seeing the descriptions to each of the organelles coming up. So let's just show you first what they visually look like, okay? And then we'll go through some of the functions. So remember, chemistry and molecules and carbons and oxygen and hydrogen and nitrogen and carbohydrates, proteins, fats, nucleic acids, they all come together and they form the cell. And then the cells come together and they adhere and they make tissues. Cells come together and they come together by cell junctions, which you will learn about in lecture. You will learn about tight junctions and gap junctions and desmosomes and hemidesmosomes and adherens junctions. These are all ways of holding cells together. And of the 4, epithelium and connective tissue take the longest time to go over, so you want to stay ahead of the curve and prepare. Muscle and nerve are pretty quick and easy. It's epithelium that has many subcategories, and it's connective tissue that has many categories, and then categories under the categories. So please prepare and time manage for that bilayer. So when you get into chemistry and lecture, you will learn a little bit about the phospholipid bilayer. Then we move inward and we have this clear translucent liquid in here, which is called a cytosol. The clear liquid is the cytosol. When we talk about the liquid, when we talk about everything from the cell membrane inward to the nucleus and the nucleolus, everything within there is referred to as the cytoplasm. The cytoplasm is really the cytosol with all of these organelles combined, right? And that's what these are all referred to as, organelles. They sound like small organs, right? Organelle. So we have the plasma membrane or cell membrane, and that is a phospholipid bilayer. It's 2 layers thick. As we move in, we have the cytosol, which is that clear liquid. And let's go to the most obvious. Let's go to that large structure in the center, the very, very large ball, right? If we look at the clear translucent region, that is called the nucleus. But then we see this orange colored pit that's in the center of that, and that is called the nucleolus. So you have a nucleolus, and then you have a nucleus. They're not the same thing, different structures, different functions. Then outside of the nucleus, we have this white membrane that is called the nuclear envelope. That's called the nuclear envelope. And these little holes are called nuclear pores. Nuclear pores. So you have nucleolus, nucleus, nuclear envelope, and within the nuclear envelope, the nuclear pores. Each of these have specific functions. Now we have this green-like membrane that is just outside of the nuclear envelope. And if we look at that, this entire structure is called the endoplasmic reticulum. The endoplasmic reticulum. But there are two types of endoplasmic reticulums. If we look off to around the 3 o'clock position here, you see it says smooth endoplasmic reticulum. And then around the 4 o'clock position, you see it says rough endoplasmic reticulum. The difference between the smooth and the rough, if you look at the structure, the smooth ER looks smooth. The rough ER looks very rough because these white speckled dots on top of them are referred to as ribosomes. Ribosomes. Ribosomes are these white speckled dots. Some of those ribosomes are fixated to the endoplasmic reticulum. When they're fixated, we call it a rough ER. There are ribosomes that could be freely floating in the cytosol, and then we would just refer to those as free ribosomes, and they would just look like white speckled dots. Those are free ribosomes. All right, what else do we have located within the cytoplasm? There is this yellow structure around 5 o'clock position. That yellow is called a lysosome. We'll talk about their functions in just a moment. We have something that looks like it and sounds like it, but it's this gray one here called a peroxisome. So lysosomes and peroxisomes sound alike, a little bit different. This one is a little bit larger, it's yellow. This one is gray, a little bit smaller. We'll talk about their functions again in a moment. Then we have the black circle that's called a centrosome with these yellow-like spindles, these cylinder-like structures called centrioles. The yellow are centrioles and the black is the centrosome. You will hear these centrioles in terms of function when we talk about mitosis and cell division. You will see these centrioles come into play. Here we have this other structure. It looks like it's, I don't know, pinkish over here, reddish, pinkish. It's called the Golgi Apparatus or the Golgi Complex. The Golgi Apparatus or the Golgi Complex. The vessel that's right here, this one here is a secretory vessel. It's not labeled, but on another image it will be. It's a secretory vessel. The orange-like structure is the mitochondria. Mitochondria. All right, I think we covered all the main structures of the cell. Let's look at some of their functions.
I have a question, Professor.
If you can hold them to the end, that would be great.
Oh, sorry.
The cell membrane is the outer boundary of the cell. That's what we showed you before. It protects the contents of the cell. It controls the movements of substances into and out of the cell, and it's semi-permeable. Semi-permeable means that it can allow things in and out, but it's selectively permeable due to the lipid bilayer. I said earlier it is a phospholipid bilayer, and it's made up of proteins, and it's made up of lipids. Lipids could be fats, and proteins are proteins. But proteins are really, really important. They're like the gatekeeper of what is permitted in and out. When you talk about the channels or protein channels or sodium channels and calcium channels, these channels are doorways that allow things in and out of the cell. They're made up of proteins. The fluid outside of the cell is called extracellular fluid. The cytoplasm are the contents of the cell, right? Everything going from the cell membrane, the inside of the cell membrane, to the nucleus, but outside of the nucleus, that's all cytoplasm. The cytosol is the internal fluid of the cell, whereas the fluid outside the cell is extracellular. There are some cells have microvilli, and I'll show you what those look like. Microvilli and cilia are similar. Cilia are larger, finger-like extensions. The microvilli are much, much smaller finger-like extensions that come off of the cell. And the microvilli are designed to increase the surface area of the cell, and it also allows for absorption, for great absorption of materials if it wants to take things in like a sponge into the cell. The cilia, these can whip around, these can move and help mobilize the cell, and these can extend off of the cell membrane. And you can also find the cilia along the respiratory tract. You can also find the cilia along not just the respiratory tract, but even the reproductive tract, especially the female reproductive tract. Organelles are everything that we've been talking about. These are all the different structures within the cell. They have different structures, they have different functions, and they're all involved in maintaining the healthy cell function, cell growth, cell maintenance, and repair and reproduction. The centrioles, these are paired organelles. Those are those yellow-like structures that are composed of microtubules. Microtubules are made of a protein, and this protein is called tubulin. Okay, that's kind of neat. And then they form this spindle apparatus. And the spindle apparatus we will see heavily involved with cell division. Cell division is what we refer to as mitosis. The centrosome, that is the dense body near the nucleus of the cell that contains the centrioles. That was the darkened black circle. The cytoskeleton is a network of protein elements throughout the cytoplasm. Okay, the cytoskeleton really just helps to maintain the structure of the cell. Now, you heard me mention ribosomes as either being fixed ribosomes or free ribosomes. Ribosomes, these are complexes of RNA and complexes of protein that are found in all cells. And the function of ribosomes is they're involved in the synthesis of proteins. Synthesis means the production of proteins. And these ribosomes come from the nucleolus. Remember, there was the nucleus, and then that orange pit deep called the nucleolus. So the nucleolus is where these ribosomes come from. The fixed ribosomes, these are the ones that are attached to the ER, to the endoplasmic reticulum. And these are going to make proteins that are used for secretion out of the cell. So it can make proteins that are needed to be exported out, or those proteins that are produced there could be used to recycle any damaged protein within the cell membrane. Remember, I said the cell membrane is made up of proteins and fats. So the proteins first enter the endoplasmic reticulum, where they are modified and they are packaged in a specific way for secretion. The free ribosomes, these are not fixed to the endoplasmic reticulum. These are just floating around in the cytoplasm. And these produce proteins that are needed for the cell itself, not necessarily to be exported out, but really to be utilized by the cell. The nucleus is the control center of the cell, which is where your genes are located, and it contains DNA. DNA is deoxyribose nucleic acid. Deoxyribose nucleic acid. That's what DNA stands for. And the DNA has the blueprint. It has the instruction manual for what proteins need to be produced. Okay, they're not producing the proteins, they just contain the instructions as to what proteins the body needs. It is the nucleolus, which is the site of the ribosomal RNA synthesis, that's involved in the actual production of the proteins. So the nucleus has the instruction manual, it has the codes, but the nucleolus carries out the duties of actually making them. Okay. So the endoplasmic reticulum, remember, has the rough ER and the smooth ER. The rough endoplasmic reticulum is continuous with that nuclear envelope. And the rough endoplasmic reticulum is involved in producing proteins, glycoproteins, and phospholipids. So you have to get comfortable with some of these words because you're going to be hearing them throughout the course. Not just proteins, but phospholipids. You're going to hear about them quite a bit, and glycoproteins. The smooth endoplasmic reticulum, it's continuous with the rough ER, but it's not involved in making proteins. This one is involved in carbohydrate metabolism and with the synthesis, with the manufacturing and the modification of lipids. Lipid is a type of fat. Okay, so I also would like to go over the other PDF that has the cell. So let's take a look at some of these structures. So again, right, same model, maybe some slightly differences here and there, but a little bit more is labeled here. So here's the cell membrane, that's the outer part. The black is the centrosome, whereas the spindles themselves are the centrioles. Remember, the clear part in the center is the nucleus, with the pit being the nucleolus. The white covering is the nuclear envelope, and then these holes that we see on the nuclear envelope are referred to as nuclear pores. Those are the holes in the envelope. Continuous with the nuclear envelope is that endoplasmic reticulum, and we can see that these have the white speckles on them. So this would be the rough endoplasmic reticulum. But this section here that does not contain the ribosomes are referred to as smooth. Here's another one. You could see that here, this is the smooth endoplasmic reticulum. There's no ribosomes here, but there are ribosomes here. The yellow is the lysosomes. What the lysosomes are doing is that they're involved with protecting the cell. Let's say a bacteria entered the cell, or some pathogen or microbe. What the lysosomes do, these vacuoles, if you would, contain hydrogen and other digestive enzymes. And what they do is they migrate towards that invader or the pathogen, and they open up a little bit and they release these hydrogen ions. They release hydrogen and they dump lots of that onto that pathogen. And what hydrogen does, whenever you add hydrogen into a solvent, into a liquid or a solvent, let's say water, you change the pH of the water and you lower the pH to make it acidic. So when you look at the pH scale goes from 0 to 14, and 7 is in the middle. And anything from 0 to 6.999999999 infinity, right? That is an acid. 7.0 is neutral, and everything above 7.0 is an alkaline or a base. So whenever you start dumping hydrogens into something, you make it acidic and you could kill it. Think of what happens if your finger touches muriatic acid or hydrochloric acid. You can burn the skin off of your finger. So that's what lysosomes do. Peroxisomes are a little bit different. The peroxisomes are highly found in like the kidneys, and they're involved in detoxification of alcohol and drugs. So you may find them in the kidneys, and you can also find them throughout the liver. So when we talk about detoxification, we're talking about peroxisomes. Lysosomes, on the other hand, are dumping hydrogens into that pathogen or microbe to try and kill it by making that environment very acidic. Okay, the clear liquid is the cytosol. The pancake, the stacked version of the pancakes here, that is the Golgi complex, the Golgi apparatus. Now, what that does is it takes the proteins that are produced and it can repackage them and ship them out. So these little orange circles here were released from the Golgi complex or the Golgi apparatus, and there's different proteins in here. And maybe one of these proteins is going to move towards the cell membrane surface, and it's going to be used to recycle some damaged proteins to repair the membrane. It's constantly being attacked. Maybe one of these is migrating towards the cell membrane, and it's going to open up and release its protein or its contents outside of the cell. And maybe that protein that it's releasing outside of the cell, maybe it's an enzyme. Maybe it's a digestive enzyme, because enzymes are proteins. Maybe it's a neurotransmitter, which are chemical messengers. They're also proteins. And what I want you to see at about 11 o'clock right here, that looks like a light bulb. This light bulb started out to be one of these vesicles, and it moved to the cell membrane, and you could see that it's opened up. It looks like it's opening up, and it is a secretory vessel that can release its protein to outside of the cell. That method of releasing something is called exocytosis. The method of taking something into the cell is called endocytosis. So exo, like exit, it's going to release the proteins outside of the cell. And endocytosis is taking in certain products or proteins into the cell. So again, the Golgi complex is going to take the proteins and it's going to say, hey, you know what, we got to repackage this a little bit. We need this protein to go outside of the cell because we need some neurotransmitters. We got to ship that out. Hey, you know what, we got some part of the cell membrane that's damaged. Let's package this protein a little bit different. Let's ship it out and send it to, the 3 o'clock position of the cell membrane to fix up damages to that part of the cell membrane. All right. And then we have the mitochondria. The mitochondria resemble bacteria, but they are responsible for helping us produce energy. And the energy currency of the body is called ATP, adenosine triphosphate. And in order to produce energy, we need glucose and we need oxygen. So we breathe in oxygen through our nose. We take that oxygen in, and it's going to break down glucose, and it's going to oxidize that glucose. And within the mitochondria, there is this process that's called the Krebs cycle, named after Hans Krebs. And the Krebs cycle, the guy won a Nobel Prize for it. The Krebs cycle, also known as the citric acid cycle. It is also known as cellular respiration. The Krebs cycle takes place within the mitochondria, and it is part of the process of creating energy. The Krebs cycle makes some energy, but it makes a lot of coenzymes, really. And those coenzymes are used for another phase that takes place in the mitochondria that is called the electron transport chain. Sometimes it's abbreviated as the ETC. ETC, again, stands for the electron transport chain. And that is dependent on iron, and it's dependent on coenzyme Q10, and it's involved in B vitamins, and involves manganese and magnesium. A lot of vitamins and minerals are needed to produce energy. And it helps to explain why when people are iron deficient or they have iron deficiency anemia, they have no energy. They're fatigued and they're tired all the time because the mitochondria are really, really dependent on iron to function. Okay, and because the mitochondria resemble bacteria, it helps explain why when someone's on an antibiotic, they tend to be tired because the antibiotic tends to destroy bacteria and anything that resembles it. They tend to target mitochondria. And most mitochondria in the body are found where most energy is needed. So if you think about what parts of the body are utilizing energy a whole lot, it's muscles, it's tendons. Muscles are constantly contracting. It's the heart. The heart is always beating. It's the brain. It's always thinking and sending out messages. So those parts of the body are highly concentrated with mitochondria. And it helps to explain why people who take certain classifications of antibiotics sometimes get muscle and tendon damage. There are certain forms of antibiotics that people tear the Achilles tendon very easily, not from doing anything stressful, just from walking and they hear a snap. So it's always important to look at the antibiotics, especially the quinolones, to make sure that there are no tendinopathies that can occur as side effects. And you have to be really careful with tendinopathies, secondary to mitochondrial damage. Mitochondria are super important. They're finding them related to many, many diseases. They are called mitochondrial dysfunctions. Like Alzheimer's is a mitochondrial dysfunctional disease. Migraine headaches, mitochondrial dysfunctional disease. Heart disease, mitochondrial dysfunctional diseases. Type 2 diabetes, type 1 diabetes, mitochondrial dysfunctions. So you have to say, well, if the cell isn't producing enough energy, those cells make up tissues, the tissues make up organs, the organs make up systems. If there's not enough energy being produced, there has to be malfunction, and malfunction is disease. All right, so make sure you review those. Let's take a look. Now we're just going to blow it up into larger section. So again, here's a peroxisome. Here's the mitochondria. This is the Golgi apparatus, the orange structures. These are called secretory vesicles, which come from the Golgi apparatus or Golgi complex. The clear structure is the cytosol. That's the clear liquid. Here's a mitochondria. The black structure is the centrosome, with the centrioles are the spindles. Because these have the white speckles on them, those are fixed ribosomes. We call that the rough endoplasmic reticulum. Don't call it rough ER. There's no abbreviations that are permitted on exams. You have to write the whole thing out, rough endoplasmic reticulum. If we look in the center, the nucleus is the largest organelle, and then deep is the nucleolus. Here is the white nuclear envelope with the nuclear pores associated with it. Here's the yellow lysosome. That's what dumps the hydrogen into the pathogen to destroy it, making it more acidic. Here's the rough endoplasmic reticulum. The ribosomes are the fixed white dots associated with it. Because there's no ribosomes here, we can refer to that one as a smooth endoplasmic reticulum. Here's the yellow lysosome once again. The gray one here would be a peroxisome. Okay. Out here is the cell membrane or the plasma membrane, or some refer to it as the cell wall. Okay, so make sure you review the cell, the organelles, structure, and function. Let me close that one out. Let me show you, let's go back one page here. I do have a video here if you'd like to review it on the cell, and it goes over the organelles and again, structure function type of thing. And then you can self-test yourself. So that's what's there for you as well. Let's just watch this 4 or 5 minute video here on cell mitosis.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the 2 sets of chromosomes. Mitosis is now complete. After mitosis, 2 new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of 3 stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes.
Microtubules.
These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This
In the middle,
occurs
they're
during
metaphase.
anaphase through a shortening of the microtubules attached to the
Shortening
kinetochores.
pulls them away. Watch.
Additionally, the poles of the cell move farther apart,
Forming the letter A.
causing increased
for anaphase.
separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the 2 ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
Great. Okay, sorry about that slight interruption there. Okay, let me close out that PowerPoint. Okay, so now that we've done that brief video on mitosis, let's take a look at the phases. Now, you can put this little mnemonic PMAT, right? PMAT, P-M-A-T. Those are the main phases where you have prophase is the P, M for metaphase, A for anaphase, and T for telophase. And what's happening in that pause is interphase, right? It's happening after it's completed and it's done. So you have PMAT, prophase, metaphase, anaphase, telophase, and then interphase. So if we look here, I think it's just that first one. Yep. Let's take a look at this first one, because here we have several of the phases showing up in sequential order. So here, number 1, number 2, and number 3, all three of these are prophase. They just break it down and say, well, here's early prophase, here's middle prophase, and here's late prophase. But after late prophase, we start to see these lining up in the center, which is metaphase. That's number 4. Then we have number 5 and number 6, where in number 5, those spindles are now starting to shorten to pull the chromosomes to their separate poles. So number 5 and 6 are both anaphase. This is just early anaphase, and this is late anaphase. Then we start.
Sorry, Dr. Linder. We don't see it. Are we supposed to see a picture?
Oh, hold on. Yep, give me a second. Okay, so thank you for letting me know. Let's go back here and look at PMAT. So you have prophase, metaphase, anaphase, and telophase. PMAT, P-M-A-T. Interphases at the very end. So here, number 1, 2, 3, 4, 5, 6, 7, 8. So they're going to go in sequential orders. So numbers 1, 2, and 3, 1, 2, and 3, these are prophase. Number 1 is early prophase, middle prophase, and 3 is late prophase. Now we start to see the chromosomes lined up in the center, in the middle, where this is going to be metaphase. There's only one metaphase, number 4. Then we start to see number 5 and 6, where the spindles start to shorten and they pull the chromosomes to their opposite poles. So this would be early anaphase, and this would be not late anaphase, but just anaphase. You have early anaphase and anaphase. So far, we have early, middle, and late prophase, metaphase, early anaphase, then regular anaphase, and then that's PM, A, now we have T for telophase. We could see the invagination right here beginning. It's starting to separate to kind of do something like this, where it wants to invaginate like this to come in, and then eventually they separate and you have interphase, which is number 8. Okay, so on an exam, all you'll have to do is really just identify the phase. I'm not going to be asking you the structures deep within to know which is the aster or which is the spindle or the chromatid. I really just want you to know PMAT, prophase, metaphase, anaphase, telophase, and of course, interphase, pure identification. In your lecture portion, they may hold you responsible for a little bit more of the description as to what's happening in each phase, but for the lab, it's more just pure identification of the phase to recognize it. Okay, so again, if you want to rewatch that, when you get into the exam 1 folder, before you click on that top folder, you can see there's the mitosis video that we watched in class just now. Then you can review the cell. And then if you want to test yourself on organelle structure and function, you could watch these 2 videos here. Otherwise, when you click back into that folder, we've already done the cell model, and we did the mitosis. So now what I'd like to do is to move a little bit ahead and just cover bone histology, muscle histology, and skin, these 3, to keep you a little bit ahead of the game. Now remember, there are 4 types of tissue. So after the cell, we finished the cell, we did the cell organelles and some of the functions of the organelles. When cells come together, they make tissues, and there's 4 types. Of the 4 types, there's epithelium, connective, muscle, and nerve. Next week, we'll do epithelium and connective. Today, we're going to introduce a little bit of muscle. We'll knock that category out. And maybe even nerve. I'll talk a little bit about nerve. And I will talk about connective tissue a little bit in relationship to blood, because blood is a type of connective tissue. It's just liquid connective tissue. And we'll talk about bone, which also falls under connective tissue. Okay, so just to give you an idea as to the direction that we're working in. Remember, epithelium and connective, there's a lot to it. So when you have the time and you can make a little bit of time, start moving ahead to the epithelial lecture that I did in the connective tissue. Okay, it's only going to help you. All right, so let's look at the muscle, because this one's pretty short. There's only 3 types of muscle, and it's cardiac muscle, it's skeletal muscle, and it's smooth muscle. And cardiac muscle and skeletal muscle are both striated in appearance. If we look here, we can see a bunch of linear striations, a bunch of lines. Some are going vertical, some are going horizontal. but it looks striated. When we look at skeletal muscle, this too looks striated, looks like a grid. So skeletal muscle and cardiac muscle both have a striated appearance. But when we look at smooth muscle, on the other hand, smooth muscle is smooth. There are no striations involved. When we look at skeletal muscle, what gives it its striated appearance is actin and myosin protein filaments. And as light passes through the microscope of those protein fibers, it gives it a very linear striated type of appearance. And the same thing is true with cardiac muscle. The way the light passes through the protein fibers, it gives it a very striated type of appearance. But
I'm sorry, what does striated mean?
Striations,
Yes.
lines.
Okay,
Like
thank
think about looking at Venetian blinds. If you turn the Venetian blinds,
If you turn the blinds, right, they start to open up, and you can see how the light moves through the blinds, very striated in appearance. The other significant difference between cardiac and skeletal is cardiac muscle is involuntary. You can't consciously control it. Your heart beats on its own. You can't tell it to beat faster or slower. But you can tell your skeletal muscles, the muscles that attach to your skeleton, to move. I could say, okay, arm move up, arm go down, fingers open up, fingers close. That's all voluntary. So skeletal muscle is voluntary, cardiac muscle is not. The other difference between cardiac muscle and skeletal muscle is that cardiac muscle is branched. You can see that these have different branches, almost like, let's say here's the tree trunk, and then there's branches that comes off of it. They divide. That's one difference between cardiac and skeletal. Cardiac is more highly branched. And then we have these purplish lines. These are called intercalated discs. Each of these black lines are intercalated discs. And all intercalated discs is when you bring one cardiomyocyte together and another, where they come together, there's this line that runs perpendicular to it. It's called an intercalated disc. And if we were to open up the intercalated disc and separate them and look at them inside, you would see what are referred to as gap junctions. And when you get into a lecture and you talk about the type of junctions that hold cells together, one of them is called a gap junction. And gap junctions are linked to different types of tissues where fast, rapid forms of communication are needed. So one heart cell must communicate with another heart cell rapidly to let it know, hey, we're going to beat. Okay, right atrium and left atrium, let's beat in harmony. So these cells have to communicate in a super rapid way. So that's the cardiac muscle. This is skeletal muscle. Smooth muscle is found around organs that you can't voluntarily control. It's also involuntary. You find it around the woman's uterus. You'll find it around organs like the intestines. You'll find it around blood vessels. Smooth muscle allows things to constrict or dilate. So if this is a blood vessel and they constrict, your blood pressure goes up. And when the blood vessels dilate, your blood pressure goes down. If this was your colon and your colon constricts by the smooth muscle, you're constipated. If the smooth muscle opens up, you can have loose stools. If it's around the trachea or your windpipe and it constricts, you have asthma. When you're exercising and they dilate, it allows you to get air in much more rapidly. Okay, so I would just know the 3 different types of muscle: cardiac, skeletal, and smooth. The only one that's voluntary that you have conscious control over is skeletal, yet skeletal and cardiac are both striated in appearance. Smooth does not have any striations by its name. It is smooth. Smooth is found around organs like the stomach and the intestines and the uterus and your blood vessels. Involuntary. Let's look at the bone histology. We'll look at that one next. Okay, so it looks like this. Now, bone comes in 2 different flavors, right? There's spongy bone and then there's compact bone. And when you look at long bones, there's different classifications of bones. You will learn about short bones like the carpals and the tarsal bones. You'll learn about irregular bones. You will learn about sesamoid bones. You will learn about long bones. Now, long bones would be like the humerus and the radius and the ulnar and your fingers, the phalanges, your femur, the tibia, the fibula. Those are very, very long bones, but long bones have unique characteristics to it. They all have a proximal and a distal epiphysis, which are the knobby parts of long bones. And then they have a shaft in the middle. So they both have a proximal and distal epiphysis with a shaft. And the proximal and the distal epiphysis are spongy bone. The shaft is compact. And when we talk about compact bone has these very unique structures that are called osteons. You see these, an osteon is like, envision a tree that was chopped down. And when you chop down a tree, if you look at what's left at the stump, it has all of these concentric rings to it. So all of these concentric rings that you're looking at, they're only found where you have compact solid bone, and they're referred to as osteons. So this entire ring-like structure, that's an osteon. All of these that look like a wedding cake, all of these is another osteon. This entire wedding cake is an osteon. All of these are different osteons. All of these are osteons. Okay, see if we could show you another one. All of these are individual osteons. Now, there are different parts to each osteon. So let me show you what some of those parts are. Also with bone, when we're building up bone, you're going to learn about different types of bone cells. There's an osteoblast and an osteoclast, and they work in harmony together. The osteoblast is building up bone, and the osteoclast is breaking down bone. You build and you break. You build up and you break down. You build up and you break down. Osteoblast with a B is building up bone. Osteoclast with the C, for see you later, it breaks down bone. So when you make bone and break down bone and it's in a beautiful harmony to one another, we call that healthy bone remodeling. Bones can remodel just like you remodel a home. If you want to remodel a kitchen, you have to bring in the demolition team, break down the old kitchen, and then you bring in a construction team and they build up a new kitchen. So the demolition team is the osteoclast. You break down first, and then the osteoblasts are going to lay down new matrix, new proteins, new bone. So you make it, you break it. That's with everything, right? You make new hair cells, you break it down. You make new skin cells, they get broken down. You make new red blood cells, they get broken down. You make nails and then they get broken down. You make bone and then they get broken down. It's constantly being remodeled. It's normal. Okay, so that gives you a little bit of background to bone physiology and bone remodeling. When your osteoclasts are working much faster than your osteoblasts, then the bones are breaking down faster than building up. We call it osteoporosis or osteopenia. Bone is highly vascular. There's blood vessels that go to it. You can see right here at about the 3 o'clock position, there's a nutrient artery. This is the outside of the bone. This is the inside of the bone. And the outer portion of long bones are called the periosteum. Remember, peri means outside. So we have the periosteum. The innermost part of the bone, where it starts to get spongy, that's the endosteum. So there's a periosteum and an endosteum. But notice the nutrient artery breaks through the periosteum and it goes all the way through the bone, and then it runs in harmony to each osteon. And we need to bring nutrients. We need to bring calcium. We need to bring magnesium. We need to bring minerals and store them in bone. When bones break, they do bleed, right? It can be very painful because bones are highly innervated and they are vascular. So we have osteocytes. Now, remember I said you have osteoblasts and osteoclasts, and then we have osteocytes. So an osteoblast makes bone. So if I'm an osteoblast, I'm an osteoblast, I'm going to produce matrix. I'm going to produce calcium and calcium salts, and I'm going to produce the cement around me that's going to make bone. I'm the osteoblast. I'm making this matrix. I'm making bone. Well, what's going to happen to the cement eventually? Does it stay soft forever? No, it hardens. So what's going to happen to the calcium that I produce, that calcium matrix that me, the osteoblast, produces? Eventually, it's going to harden. And I, the osteoblast, I'm going to get trapped in the matrix. And I can't get out. I'm stuck within itself. I now become useless. I once was an immature cell. I was an immature cell called an osteoblast. I've made the matrix. I've done my job. The matrix and calcium hardened. Now I become useless. I now become a mature cell called an osteocyte. So an osteoblast becomes an osteocyte when it gets trapped in its matrix. And now I'm stuck. I'm stuck in this little cavity. I'm in this small little cavity, and the cavity that I'm stuck in is called a lacuna, which we'll see shortly. So these are osteocytes, and they're stuck in a little cavity called a lacuna. Each of these rings is just called a concentric lamellae. Each ring is a concentric lamellae. The lamellae that aren't concentric, they're called circumferential lamellae. I'll show you another view of that. So here's a bird's eye view. Here, each of these are a concentric lamellae. There's all concentric lamellae. And then remember this blood vessel here? That nutrient artery that broke through, and then you see how it runs through the center. It runs right through the center of each of these osteons. That center is here, here, here. All of these holes, you can see the blood vessel there. That's called the central canal, also known as the Haversian canal. So blood vessels travel through there. Here's the periosteum. That was that outer layer. That's where the nutrient artery perforated through. And all the way on the inside, on the left-hand side, that's the endosteum. And again, all of these concentric rings, all of them make up the osteons. Osteon, 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 osteon. Now, what we have in between these completed osteons, if you look here, these are circumferential lamellae. These are outer circumferential lamellae. They don't make the complete circles. These are remnants. See these here? These are remnants of old osteons that were broken down by osteoclasts. They don't make the full circle anymore. They're just remnants due to the osteoclasts. So we have the periosteum, outer layer. This is where your tendons attach, and this is where your ligaments attach to bone, to the periosteum. These are your outer circumferential lamellae. These are the osteocytes. This is your nutrient artery. These here, right, these are your concentric lamellae. These, again, that don't make those full circles and that are in between the osteons, they're called interstitial lamellae. They're interstitial. They're not concentric lamellae. They're interstitial. Like interstitial fluid, if you have one cell and another cell, the liquid in the cell is the cytoplasm. That's called intracellular fluid. But between these cells is called interstitial fluid. That's between the cells. So between each of these osteons are interstitial lamellae. They are remnants of old osteons that are broken down. All the way around the periphery, these are circumferential lamellae. Again, a close-up view. These are concentric lamellae. These are these red lines here going around. These cavities, right, these hollowed out cavities that are empty, that's the lacuna. But what sits in the lacuna are the osteocytes, these. Now remember, what was an osteocyte? It's just an osteoblast that was immature, that produced matrix. The calcium calcified or ossified, and the osteoblast got stuck within its matrix. And now there's a cavity around it. The cavity is the lacuna. The osteoblast has now become a mature cell called an osteocyte. Here's your central canal or your Haversian canal. That's where your blood vessels travel. Not just arteries, but veins and nerves and lymphatic vessels. They all go through the center of the osteon. Here again, right, this looks like the tree that was cut down. So number 3 in the center is your central canal or Haversian canal. Number 4, these are the concentric lamellae. Number 2, those are the remnants of old osteons that were broken down. Those are called interstitial lamellae. And then the cavities, those are called lacuna. What sits in there? Those are the osteocytes. Now, the osteocytes, these cells, have these finger-like extensions coming off of them, these little spider-like legs. And those spider-like legs are referred to as canaliculi. They're like little canals called canal or canaliculi. Let's see if I can show you the spelling of that somewhere here. Let's see if we can find the canaliculis. There we go. So if we look at this one, again, here's a great close-up view of just one osteon. In the center, we could see a nerve, we could see an artery, we could see a vein, we could see the lymph. So this is your Haversian canal or central canal. The hollowed out cavity is the lacunae. What's in the center, the blue, that is the osteocyte. And then the red spider-like, jellyfish-like extensions coming off of them are called canaliculi, right? It helps to communicate with other cells to figure out what's happening in the bone. It's like a matrix of communication. That's the canaliculi. What's not making up the concentric lamellae, those are called interstitial lamellae. They're just remnants of old osteons. Okay. Same thing here. We could see a vein, we could see an artery, we could see a nerve, we could see a lymph. These are concentric lamellae, each of these, whether it's the pale or the yellow rings. The lacunae are the cavities. The osteocyte is the blue, and then the red finger-like extensions are the canaliculi. These are interstitial lamellae. These are concentric lamellae. Okay. Again, same thing, nothing new. Lacunae are the empty cavities. The cavities is where the osteocyte would sit. The canaliculi are the red extensions. The blue little circle, that's just the nucleus of the osteocyte, and the red is the body of the osteocyte. No big deal. Okay, so that's bone. That's the bone histology. And again, osteons are only found where there is compact, solid bone. If it doesn't have that concentric appearance, it's not compact, then you're going to be looking at spongy bone that looks like a sponge, very porous, not as solid. Okay, and I think, where's blood? Is there blood here somewhere? Blood. Okay. In terms of blood, blood is involved in carrying oxygen and carrying carbon dioxide. It's going to distribute nutrients to all parts of the body. It's involved in carrying hormones. It's transporting waste products. It's involved in regulating pH. It's involved in immune system or your defense mechanisms of your body. It's involved in regulating body temperature. Blood is really important. When blood is drawn from your arm, right? Let's see if I can find that picture where they do that. I thought I had one like that. No? Oh boy. Okay. So when there's a venipuncture and blood is taken from your arm and it's spun in a centrifuge, it separates the blood into different parts. It will separate it into red blood cells and white blood cells. So red blood cells look like this. They're also called an erythrocyte. You can see they're biconcave, meaning it's concave on both sides, top and bottom, biconcave. There's no nucleus. Red blood cells do not have a nucleus because nucleus is like the largest organelle. And if a red blood cell had a nucleus, then there wouldn't be any room for the red blood cell to hold on to hemoglobin. And hemoglobin is needed to bind to oxygen so that it can carry, so that the iron can bind to oxygen and carry oxygen and CO2 to and from places. So when we look at a slide, this is blood, this is your red blood cell. We can see it's concave on both sides. Each of these is a red blood cell. And what's between each blood cell here, notice there's lots of matrix. When we talk about epithelium and we compare it to connective tissue, the difference between the two under a microscope is that epithelial has lots of cells that are very closely packed together with very little extracellular matrix, very little stuff around it. But connective tissue has cells that are scattered around like we see here. And the cells have lots of matrix around it. So here, these cells are not closely packed where there's no breathing space. They're just close together, but there's lots of extracellular matrix. The matrix here that's around blood is called plasma. So this is a red blood cell. But we also have white blood cells. So what we see here are red blood cells, and we see it's hypochromic, less color in the middle. And then there's plasma, the stuff around it. But then there's something here that doesn't look anything like these. So besides red blood cells, we have white blood cells. And white blood cells can be categorized as granular or agranular. When you look at this to the right in the cytoplasm, this is granulations. This is like grains of sand in the cytoplasm. When we talk about agranular, these cells have no granules. So one type of white blood cell, which is granular, and there are 3 granular sites. One is a neutrophil, one is an eosinophil, and one is a basophil. If you look at all 3 of these, notice as I toggle back and forth, although the nucleuses look different, they all have granules in the cytoplasm, whether it's a neutrophil, eosinophil, or basophil. So the neutrophil has usually 3 to 5 heads to the nucleus. So here's 1, 2, 3, maybe 4, maybe 5 heads to the nucleus. 1, 2, 3, 4, 5 heads to the nucleus, and it's very granular. Now, neutrophils are like the first responders. They're the first responders when there's a pathogen in the body. And it moves to that pathogen, and these granules degranulate, meaning they're like lysosomes. They're going to release their degradatory digestive enzymes to break down and try and destroy whatever it is that has invaded the body. So they have these antibacterial properties to it. They can find, seek, and destroy. That's what neutrophils are. Your eosinophils, these also have granules, but notice they're a little bit bigger than this, right? These red granules are much larger. So this is another granular site, but notice the nucleus is bilobed. There's only like 1, 2 lobes to the nucleus. Historically, that's what eosinophils are. You've got these 2 lobes to the nucleus and large, bright red staining granules. Eosinophils are involved with allergies. When someone's having an allergic response and you take their blood, their eosinophil count is going to be really, really high. When someone has a bacteria, their neutrophil count is going to be really, really high when there's a bacterial issue. When there's an allergy, their eosinophil count goes high. When their basophil count is high, basophil has this large nucleus and it's dark staining blue and purple. And sometimes it's stained so dark that it's really hard to kind of see the nucleus because it's kind of gets camouflaged because they're all stained dark. Basophils are also high when there is an allergic response. Basophils are going to release histamine, right? When you have a histamine reaction, your nose gets stuffed, your skin gets itchy and red. So the basophils release histamine, whereas the eosinophil is going to release histamine ACE. Histamine ACE, anything that ends in the ACE is an enzyme. So histamine ACE is going to break down histamine. So they kind of work together. One's releasing histamine, the other one's trying to get rid of it. But they're both seen with allergies. Eosinophils are high with allergies. They're also high with parasitic infections. Okay, so these 3, the neutrophil, eosinophil, and basophil, those are granular sites. Those are white blood cells that are called granular sites. The agranular sites are these 2, lymphocytes and monocytes. When we look at lymphocytes and monocytes, notice there's no granules here. There's none. There's no granules. The basophil, eosinophil, and neutrophil all have granules. Monocytes and lymphocytes, no granules. So the monocyte, notice it's got this large kidney bean-shaped nucleus without granules. And look at the lymphocyte. The nucleus is just as big as the cell. Now, Monocytes are always in circulation, and monocytes become something called a macrophage. And a macrophage is going to be like an old school Pac-Man. If this is a little bacteria, then this macrophage is going to engulf it. It's going to eat it and get rid of it. That's what macrophages do. They engulf debris and they try and clean things up. But macrophages start out as these. They start out as monocytes. So monocytes are in circulation. They're in your blood vessels. And then your monocyte goes, hey, there's a problem here in the liver. I'm going to leave the blood vessel, go into the liver. And now it changes its name. It becomes a macrophage. Or it's going to leave and it's going to go into the skin tissue to clean up debris. And now it becomes a macrophage of the skin. Or it's going to go in the lungs and become a macrophage of the lungs. So depending on the tissues that they go into, they change names. The macrophages have different names depending on the tissues they go into. So when it's in the liver, it's called a Kupffer cell. When it's in the brain, it's called a microglial cell. When it's in the lungs, it's called an alveolar dust cell. Okay. So monocytes and lymphocytes are agranular. They're not granular. And then you have platelets. Platelets are involved in creating a blood clot. Platelets help to create a thrombosis. Okay, so if we were to look at a model like this of the different types of components, there are red blood cells, lymphocytes, monocytes, neutrophils, eosinophils, basophils, platelets, and plasma. Let's look and find the number one. Here's the number one. Those are erythrocytes or red blood cells. Number two, here and here. These are agranular, and the nucleuses are as big as the cell. Those are lymphocytes. Where's number three? Here's a number three, pretty large cell, agranular. The nucleus is like a kidney bean shaped. That's your monocyte. Number four, neutrophil. There's a little spelling error there. It's N-E-U-T. There's no R there. It's neutrophil. Number four, right here. You've got 1, 2, 3. Here you got 1, 2, 3, 4, 5 heads to the nucleus. Those are neutrophils. Those are granular. Number 5, eosinophil, right here. 2 heads to the nucleus. Red staining cytoplasm with granules. That's your eosinophil. Where's our number 6? Here it has the dark blue staining. It's granular. The dark blue staining granules, those are your basophils. Number 7, these are platelets. These little star-like things in the plasma, those are platelets. And number 8, the extracellular matrix, all the liquid and fluid around all of these cells are called plasma. So when you have cells that are scattered around and they're not closely packed together, that's connective tissue. However, when you have cells closely packed together with very little matrix or extracellular matrix, that's epithelium. So that's one of the key characteristics when we look at things under a microscope. We go, are the cells really tightly packed and there's no extracellular matrix? If that's the case, it's epithelial tissue. If there's cells here and there with lots of free space around it, then that's connective tissue. Okay, so if we look here, this one here has got 1, 2, 3, 4 heads to the nucleus. That's a neutrophil. 1, 2 heads to the nucleus. That's an eosinophil. All of these are red blood cells. This is granular. Can't really see the nucleus, but lots of dots and granules. Because it's obscured, we call it a basophil. Remember, the nucleus and the granules appear as one. You can't really differentiate them too well. That's a basophil. But here's your kidney bean-shaped nucleus. Pretty large. There's no granules in the cytoplasm. That's a monocyte. Here's your lymphocyte. That's where the nucleus is almost as large as the cell. Here's 1, 2, 3, 4 heads to the nucleus. That's a neutrophil. Neutrophils respond to bacterial infections. Lymphocytes respond to viral infections. The way you can remember that is a little mnemonic: never bet Las Vegas, never bet Las Vegas, NBLV. N is for neutrophil, B is for bacteria. So neutrophil count is high with bacterial infections. And then Las Vegas, LV, L is for lymphocyte, V is for virus. So when the neutrophil count is high, you're thinking there's a bacterial infection. When the lymphocyte count is high, you're thinking there's a viral infection. Let's take a look here. These are all red blood cells. Look at number 1 and look at number 2. Let's see if we can identify these. If we look at 3, 4, and 5, I'll tell you that 3, 4, and 5 are granular sites. So that means one is a basophil, the other is an eosinophil, and the other is a neutrophil. 1 and 2 are agranular. So one's got to be a lymphocyte, one's got to be a monocyte. So remember, the lymphocyte has this nucleus that's almost as large as the cell. The monocyte has the nucleus that looks like a kidney bean shaped, and it's much, much larger cell. When we look at this one, this has the dark staining nucleus with the granules. The nucleus is almost obscured. This is the basophil. Now we got to look at these two. Which one's more red? The red staining one is the eosinophil, which means this 1, 2, 3, 4, 5, that's the neutrophil. Let's see if we're right. Monocyte, lymphocyte, 3 to 5 heads to the nucleus, neutrophil, eosinophil, basophil. Okay, let's close that out. And only one thing left. The only thing left is skin. Now, we usually save skin for last. And the reason why we would save skin for last is after you do epithelial tissue, and after you do connective tissue, and after we've done muscle, and after we've done nerve, we would go into the skin, because skin is made up of all 4 types of connective tissue. It's made up of all 4 types of tissue: epithelium, connective, muscle, and nerve. Okay, so I want to show you the different parts of skin, and then we'll go over it again. If there is time, and we go over epithelium, and then we go over connective, and we go over nerve, and there's still time, I'll go back to these and just review it, because it'll make sense a second time. So this is skin. This is a microscopic view from the top layer here, and then we start to move deeper to the bottom layer. So if we look at number 1 to the left at around Roman numeral 1, Roman numeral 2, Roman numeral 3, this upper region is the epidermis. The middle region here is the dermis. And then layer 3, where we see all this yellow fat, that is the hypodermis, or subcu, or subcutaneous. So there's the epidermis on the top, dermis that's deeper, number 2, and number 3 we can see is the key here off to the right. This is the subcu, or subcutaneous, or hypodermis. Now, under a microscope, layer 1 comes in about 5 different layers. They're called stratum layers, like stratosphere. Stratum means layer. There's a stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and then a stratum germinativum, aka sometimes called number 7, the stratum basal, the basal layer. And the way you can remember that is a little silly mnemonic. That's Cher loves getting skin Botox. Cher loves getting skin Botox. Now, Cher has been very open about all her cosmetic surgery and stuff like that, so it's not being done teasing her or making fun of her. She's done numerous interviews of how she's tried to maintain her youth over the years using Botox and injections and plastic surgery. So share for corneum, loves, lucidum, getting, granulosum, skin, spinosum, right? Share loves getting skin. Botox is number 7. The other name for germinativum is basal, like basal cell carcinoma. So they call it the stratum basal layer. So all of those layers are found in number 1, which you can see here, layer 2, 3, 4, 5, 6, and 7. These are the microscopic layers that fall under the epidermis. And they're all important. But the ones that you can really see and the ones that are super important is this layer number 2, which is made up of many, many layers of epithelium. It's a type of epithelium. And these many layers are called stratum corneum. These are dead cells. These skin cells begin down here at the bottom, where it's the germinative layer, made up of germ cells that reproduce and multiply and divide. And this germinal layer pushes the skin cells upwards in this direction. And it takes about 30 days for these cells to migrate and move upward till eventually they slough off at the stratum corneum about every 30 days. But don't worry, just like Doritos, don't worry, we'll make more. And then the germinative layers down here at the bottom produce more skin cells and they migrate upwards. And in about a 30-day journey to get to the top layer, the stratum corneum, they will eventually slough off. Okay, so you should know those share loves getting skin Botox. You should know the layers, but the thickest one up here on top is the stratum corneum. It is dead cells. The lucid means clear. Not all types of skin have the stratum lucidum, but thicker skin does. The skin of your palm and the skin of your foot, right? Where you have thicker skin that comes into contact with areas that could be abrasive or irritated. If you walk barefoot, you need more protection on the feet. If you land and fall, you need to have lots of protection on the palms of your hand. So wherever there is thicker skin, you have that lucidum layer. Where you have thinner skin, you're not going to have that stratum lucidum. You'll have all the other layers but the lucid layer. The granulosum, those cells have granules that make oils. They produce oil and keep the skin nice and lubricated. And the stratum spinosum looks spiny. It looks like a spine, very spiny. All right, so that's the epidermis. When you look at the dermis itself has a few layers. It has more of a superficial layer, which is on top, number 8, the papillary layer. That's more superficial. And then it has the deeper layer, which is number 10, called a reticular layer. So the papillary is up higher. The reticular layer is a little bit lower. Within the dermis, we see that there are a lot of accessory structures, right? If you look at this, right, this is the hair, right? If you look at number 23, 23 is the hair shaft. So this is the hair shaft. What surrounds it here, right? This is your oil gland. The oil gland is called your sebaceous gland. So this is going to produce oil that moves up the shaft of the hair, and it creates this watertight seal where the shaft of the hair breaks through the skin. It kind of has some antibacterial properties too. So this is your oil gland or your sebaceous gland. Well, what about the white one next to it? Well, if this is the oil gland, what's this? Well, the white balls right here, these are the sudiferous glands. These are your sweat glands. So number 24 is your sweat pore. So sweat moves up and is released through the skin through here to cool off the body. So what is this? This is muscle, right? Remember we said the skin has all 4 types of tissues. Well, it's got nerves. We can see number 1, these are nerves. Even over here, number 12, this is nerve tissue. We have muscle tissue. This is called the erector pili muscle right here. So when this contracts, when this muscle contracts, it's going to pull the hair on end and give you goosebumps. So this is muscle. This is nerve. Then you look, you got blood. That's connective tissue. You have epithelium. That's tissue. You have fat. That's connective tissue. So you have epithelial tissue up here. You got connective tissue down here. You got blood, which is connective tissue. You have muscle. And you have nerve. That's why we usually cover skin last, just to summarize the 4 different types of tissue: epithelium, connective, muscle, and nerve. So number 1 here, number 1, these are called Meissner's corpuscles. These are type of nerve fibers. These are for like pain and very, very light touch. When you look at number 12, this is called Pacinian Corpuscle. This is for deep pressure, and that would make sense, right? This is more superficial, so that's light touch, but these are deeper, so you need deep pressure to feel here. And these are just free nerve endings. These are just free nerve endings. These are Meissner's corpuscles. These are Pacinian corpuscles. These are just free nerve endings. But either way, they're all sensory. These are all part of the nervous system. So this is nerve tissue. This is muscle tissue. Epithelium is up here. This is epithelial tissue. You'll understand that by next week. And then connective tissue, blood is connective tissue, and fat is connective tissue. Okay. Same structures, just a little bit of a different model, but everything else is the same here. If we look on the right, this number 1 here on the right, that's your epidermis. Number 2, this layer with all these accessory structures in the middle, that's your dermis. And then down here, number 3 is your hypodermis or subcutaneous. Here, the white sac right there, that is your sweat gland, which is your sudiferous gland. This is the same thing as this, right? This is just whole, this is sliced or cut through. Here, this purple and this purple, they're the same. This is whole, this has a slice through it. So this is your oil gland, known as your sebaceous gland, and this is your oil gland, known as your sebaceous gland. See number 1, sebaceous. Here's your erector pili muscle. That's number 4, erector pili muscle. There's your Pacinian corpuscle, number 7. Here's your number 5, your Meissner's corpuscle for light touch. Number 14, the yellow, that's your fat or adipose cells. The uppermost layer, number 25, that would be your stratum corneum. Stratum corneum. And then the deepest layer looks like number 20, that's your basal layer, your stratum basal. Basal layer, number 20. Okay. Okay, the other 5 layers are here. Remember, Cher loves getting skin Botox. Then the upper part of the dermis is the papillary region, and then the lower part is more reticular. So number 17 is the reticular region. Okay, I think that was the last one I wanted to cover. Let me close that out. Let me stop the screen share. And let me stop the recording.